Welcome back to Clinical Hematology Reviews. Today we'll be discussing RBC poikilocytes. Poikilocytosis is the general medical term used to describe a nonspecific variation in RBC shape. Evaluating these specific shapes on a peripheral blood smear requires semi-quantitative grading across 10 oil immersion fields under a 100 times objective. We record the finding as slight when observing 1 to 2 distinct abnormal shapes per field, moderate when seeing 3 to 4, and marked when noting greater than 4 distinct abnormal shapes per field.
It is crucial to distinguish true poikilocytes from artifactual morphology introduced during slide preparation or sample handling. For instance, crenated RBCs can form in aged EDTA tubes due to hyperosmolality, which draws water out of the cell and mimics echinocytes. Mechanical shearing forces during smear creation produce mechanical ovulocytes, while a platelet resting directly on an RBC creates a pseudo-target cell. Furthermore, we should remember that altered membrane lipid dynamics dictate specific true shape alterations. Equal expansion of cholesterol and phospholipids expands total membrane area to form target cells, an increased outer to inner leaflet ratio forms echinocytes, and an expanded inner leaflet relative to the outer leaflet forms stomatocytes.
Moving to our first specific morphology, let's detail acanthocytes, which are also known as spur cells. Morphologically, these are small spherical RBCs that completely lack central pallor. They are characterized by multiple irregular thorn-like or spiky projections distributed along the membrane. These projections vary in both length and width and frequently feature small bulb-like tips.
The underlying pathophysiology stems directly from an altered membrane lipid composition. Specifically, there is an accumulation of unesterified free cholesterol in the outer membrane leaflet. This excess free cholesterol increases outer membrane fluidity, generating these asymmetric, spiky projections. Clinically, acanthocytes are strongly associated with severe liver disease, such as spur cell anemia, as well as A-beta lipoproteinemia.
Next, we have target cells, which are also termed codocytes. In physiological circulation, these cells are thin and bell-shaped. However, on a stained Wright's blood smear, they present a characteristic target or bullseye appearance. You will observe a central pink-staining hemoglobin button surrounded by a clear achromic zone bounded by an outer ring of pink hemoglobin.
This distinct morphological pattern results from an increased surface area to volume ratio within the red cell. This is caused either by an absolute excess of membrane lipids or by a decrease in intracellular hemoglobin content. Clinically, target cells are frequently identified in various hemoglobinopathies, including hemoglobin S, hemoglobin C, and hemoglobin E. They are also widely observed in thalassemias, iron deficiency anemia, and chronic liver disease.
Teardrop cells, or dacryocytes, are RBCs elongated at one end to form a distinct point, giving them a pear or teardrop appearance. One primary mechanism driving their formation is splenic pitting by reticuloendothelial system, or RES, macrophages. As red cells containing inclusions like Heinz bodies or Howell-Jolly bodies pass through narrow splenic cords, macrophages pit out the inclusion. This action stretches the cell membrane beyond its ability to rebound into a biconcave shape.
Another major mechanism occurs in fibrotic bone marrow, where dense collagen fibers replace normal stromal elements. Rigid red cell precursors are physically stretched as they squeeze out of the fibrotic marrow space into circulation. Additionally, traversing microvascular fibrin networks or microthrombi can mechanically stretch circulating cells. Clinically, teardrop cells are a classic finding in primary myelofibrosis, or PMF, as well as myelophytic anemia, thalassemias, and severe anemias.
Sickle cells, or drepanocytes, appear as elongated, crescent-shaped or banana-shaped RBCs with pointed ends. Reversible forms may exhibit rounded ends with a flattened side, whereas irreversible forms feature sharp, pointed ends. This striking shape alteration is driven by mutated hemoglobin S. Under conditions of decreased oxygen tension, reduced pH, or hypothermia in capillary beds, deoxygenated hemoglobin S polymerizes into rigid crystalline rods.
As polymerization continues within the cytoplasm, the cell shifts through an intermediate holly leaf shape before distorting into a rigid sickled cell. These polymerized red cells completely lose structural deformability and demonstrate increased mechanical fragility. Consequently, they become trapped within microcapillaries, precipitating vaso-occlusive crises, tissue ischemia, organ damage, and chronic hemolytic anemia.
Echinocytes, or Burr cells, are typically smaller than normal RBCs. They feature numerous, short, uniform, regular spine-like projections evenly distributed across the entire membrane surface. Pathophysiologically, true echinocytes develop when the surface area of the outer membrane leaflet expands relative to the inner leaflet.
It is important to recognize that echinocyte transformation is initially reversible, allowing the cell to revert back to a normal discocyte under proper physiological conditions. However, if true echinocytes remain in circulation, splenic macrophages strip away the membrane spines, reducing total membrane surface area and converting them into spherocytes. True echinocytes are associated with uremia, end-stage renal disease, pyruvate kinase deficiency, and severe electrolyte imbalances, whereas artifactual crenated cells stem from hyperosmolar changes in aged samples.
Turning to ovulocytes and elliptocytes, these cells range from elongated ovals with a wider diameter to narrow, highly elongated rod-like forms. The narrower elliptocytes are often referred to as pencil cells or cigar cells. Despite their elongated structure, both forms typically retain a distinct central area of pallor.
These abnormal shapes arise from qualitative or quantitative defects in the RBC membrane cytoskeleton. Specifically, structural defects occur in key peripheral membrane proteins, such as ankyrin and spectrin. As red cells experience mechanical shear stress traversing microcapillaries, the defective cytoskeletal scaffolding fails to rebound, causing permanent elongation. Clinically, these cells are prominent in hereditary elliptocytosis, or HE, as well as iron deficiency anemia and thalassemias.
Keratocytes, or helmet cells, display a notched concavity on one side flanked by two horn-like spicules at the margins. The primary pathophysiological mechanism begins when a circulating RBC encounters an intravascular fibrin strand. The red cell becomes impaled on the strand, folding over it on both sides.
The opposing inner membranes on the hanging sides touch and fuse together, pushing hemoglobin to one side and creating a clear membrane-bound vacuole on the other, forming a blister cell. As mechanical fluid pressure forces the cell past the obstruction, the blister vacuole bursts, leaving behind the notched concavity with two sharp spicules.
Keratocytes are key markers of microangiopathic hemolytic anemias, or MAHA, including disseminated intravascular coagulation, or DIC, thrombotic thrombocytopenic purpura, or TTP, and hemolytic uremic syndrome, or HUS.
Schistocytes are fragmented red blood cells that demonstrate a wide variety of irregular, sharp shapes, including triangular, comma-shaped, crescent, or helmet-like fragments. They are typically microcytic in size. These fragments are produced by direct mechanical trauma, such as shearing against intravascular fibrin networks or artificial heart valves.
Although these fragments retain normal intracellular membrane deformability, their overall cellular survival is severely compromised. These fragmented cells undergo rapid intravascular hemolysis or are culled rapidly by macrophages in the spleen. Clinically, schistocytes are critical findings in MAHA conditions like DIC, TTP, and HUS, as well as in patients with mechanical cardiac valve prostheses, severe burns, and vasculitis.
Spherocytes are small, densely staining, perfectly round, sphere-like RBCs that completely lack central pallor. Although they appear microcytic on a blood smear due to a reduced diameter, their cell volume, measured by the MCV, is typically within the normal reference range. Spherocytes are unique because they are the only poikilocyte classified as hyperchromic. This hyperchromia is reflected by an elevated MCHC, which frequently exceeds 36 grams per deciliter.
Pathophysiologically, spherocytes form when biconcavity is lost due to a reduced surface area to volume ratio. In hereditary spherocytosis, inherited defects in spectrin, ankyrin, or band 3 proteins cause spontaneous membrane blebbing. Splenic macrophages call these blebs, reducing total membrane surface area. In immune hemolytic anemia, antibodies or complement bind to the membrane, leading splenic RES macrophages to partially phagocytose membrane chunks and forcing the remaining membrane to seal into a sphere.
Finally, let's examine stomatocytes, also known as mouth or clam cells. These are uniconcave RBCs featuring a distinct slit-like or mouth-like central region of pallor. True stomatocytes result from an altered membrane lipid composition, where the inner membrane leaflet expands relative to the outer leaflet. Alternatively, they can stem from abnormal membrane cation permeability, involving sodium-potassium pump defects.
While true stomatocyte morphology can be reversible, it is also one of the most common artifacts encountered on peripheral blood smears. Artifactual stomatocytes frequently result from slow smear drying, acidic stain pH, or improper slide preparation techniques. When genuine stomatocytosis is present, clinical associations include hereditary stomatocytosis, acute alcoholism, obstructive liver disease, and Rh deficiency syndrome. Today, we reviewed the major red blood cell shape abnormalities and their clinical significance.
Excellent review. Thanks for joining us.